Welcome to our lecture on reproductive biology. Today, we are going to explore a truly fascinating topic, sperm transport and maturation. You might think that once sperm are produced, they are immediately ready for fertilization, but that is far from the truth. We will uncover the incredible journey these cells take through the male reproductive tract, transforming from completely immotile cells into highly active, fertile spermatozoa. Let us dive in and see how this biological conveyor belt actually works. To understand this process, imagine a massive car factory. The testes act as the initial assembly line, building the basic chassis of the car. However, right off this line, the car lacks an engine, tires, or a steering wheel. If you try to drive it, it will not move an inch. Similarly, the millions of sperm cells produced in the testes are structurally complete, but they are completely unable to swim or fertilize an egg on their own. They need to undergo a crucial finishing process to become fully functional. Now that we know the sperm need finishing touches, let us look at where this happens. These unfinished sperm must travel down a long, highly coiled biological conveyor belt known as the epididymis. This tightly coiled tube is divided into three distinct anatomical regions: the caput, or head, the corpus, or body, and the cauda, or tail. Because the newly formed sperm are immotile, they cannot swim through this tube. Instead, they are passively transported via fluid currents and pushed along by rhythmic, smooth muscle contractions in the walls of the tract. As the sperm are pushed along this conveyor belt, what exactly is happening to them? During their journey through the caput and corpus, they are bathed in special fluids. These fluids add the necessary biochemical parts for them to survive in the female tract. Critical changes occur, such as the addition of specific surface proteins and important modifications to their cell membranes. By the time they reach the final section, the cauda epididymis, they are fully mature, high-performance cells. They are stored here as motile and fertile spermatozoa until ejaculation propels them through the vas deferens. Understanding this maturation process is absolutely critical in the real world, especially for diagnosing and treating male infertility. If a man's sperm move through the epididymis too quickly, they will not have enough time to mature, resulting in an ejaculate full of live but useless sperm that cannot swim. Conversely, if there is a physical blockage in this transport system, no sperm will reach the ejaculate at all, a condition known as obstructive azoospermia. Fertility specialists use this knowledge to extract sperm directly from the testes for advanced procedures like intracytoplasmic sperm injection, manually bypassing the need for natural maturation. Since we just learned that moving too quickly prevents maturation, we must look at the time sperm spend inside this biological incubation chamber. We can model the transit time of sperm through this coiled tube using a fundamental kinematic equation adapted for biological fluid transport. The formula is T equals L divided by V average. Here, T represents the total transit time required for a sperm cell to travel through the epididymis, typically measured in days. L represents the total uncoiled length of the epididymal duct, measured in meters. Finally, V average represents the average velocity of the fluid flow pushing the sperm, measured in meters per day. Remember, this is not the swimming speed because the sperm are paralyzed during this journey. If the length increases, transit time increases. If fluid velocity increases, transit time decreases. Now that we have our formula, let us see this with a concrete example. A reproductive biologist is evaluating a patient for unexplained infertility. We are given that the uncoiled length of the patient's epididymis is 6.0 meters. The average velocity of the fluid transporting the immotile sperm is measured at 0.5 meters per day. We need to calculate the total transit time to see if it falls within the normal 10 to 14-day window. We start by writing our formula. T equals L divided by V average. We substitute 6.0 for L and 0.5 for V average. Dividing 6 by 0.5 gives us exactly 12 days. Because normal maturation requires between 10 and 14 days, this result physically means the patient's sperm transport speed is perfectly normal, and the infertility issue must lie elsewhere. We have seen how time affects the biochemical maturation of sperm. But that is only half the story. As sperm travel through the epididymis, they do not just mature biochemically. They must also become highly concentrated physically. When sperm first leave the testes and enter the epididymis, they are suspended in a very large volume of testicular fluid. In this highly dilute state, they take up too much space. This large volume is completely inefficient for storage in the male reproductive tract and would make the final ejaculate far too dilute to be effective for fertilization. So, how does the body solve this dilution problem? The answer lies in the specialized epithelial cells that line the walls of the epididymis. As the fluid carrying the sperm passes by, these epithelial cells actively absorb most of this fluid, pulling it out of the duct and back into the bloodstream. By removing the liquid while leaving the cells behind, the sperm are packed tightly together for efficient storage in the cauda region. This massive fluid absorption is a critical part of maturation, as it creates the dense sperm reserves needed for a successful and potent ejaculation. To understand clinical semen analysis reports, we can calculate this final concentration using a basic volumetric formula. The formula is C final equals N total divided by the quantity V initial minus V absorbed. Let us break down these symbols. C final represents the final concentration of sperm in the tail of the epididymis, usually measured in millions of sperm per milliliter. N total is the total number of sperm cells entering the epididymis from the testes. V initial is the initial volume of fluid carrying these sperm, and V absorbed is the volume of fluid that the epithelial cells absorb. As the absorbed volume increases, the denominator becomes smaller. Physically, this means the final concentration of sperm increases dramatically, packing them densely for storage. Let us apply this concentration formula to a laboratory analysis. We are given that 500 million sperm enter the caput, suspended in an initial fluid volume of 50 milliliters. As the fluid travels, the epithelial cells absorb 45 milliliters of fluid. We need to calculate the final sperm concentration. First, we determine the final volume of fluid remaining by subtracting the absorbed volume from the initial volume. 50 minus 45 leaves us with just 5 milliliters of fluid. Next, we divide the total number of sperm, which is 500 million, by this remaining volume of 5 milliliters. This straightforward division yields 100 million sperm per milliliter. This result physically demonstrates that by absorbing 90% of the fluid, the epididymis has concentrated the sperm tenfold, which is absolutely essential for fertility. Now that we have calculated both the time and concentration, let us visualize this entire pathway. On screen is a schematic diagram of the male reproductive tract. Starting at the bottom left, you can see the seminiferous tubules, where the structurally complete but immotile sperm are produced. Follow the arrows, and you will see them enter the caput, or head, of the epididymis. As they travel through the highly coiled corpus, or body, the critical biochemical maturation and fluid absorption processes occur. Finally, follow the pathway down to the cauda, or tail. This is where the now fully mature, highly concentrated, and motile sperm are stored. When ejaculation occurs, they will exit upwards through the straight tube on the right, labeled the vas deferens. We have covered the full topic of sperm transport and maturation, so let us review the most important ideas. First, remember that the epididymis acts as a biological finishing school, transforming structurally complete but functionally useless cells into highly motile, fertile spermatozoa. Second, this process is highly time-dependent. The sperm must spend adequate time, usually 10 to 14 days, traveling through the duct to acquire the necessary biochemical tools. Third, massive fluid absorption by the epithelial cells concentrates the sperm tenfold, preparing them for dense storage in the cauda. Without this intricate, time-sensitive conveyor belt system, natural human reproduction would simply not be possible. You now have a solid understanding of how sperm mature and prepare for fertilization.